Dr. Christelle Nilles is presenting the webinar today, and we are very grateful for the time she's given us this morning. Dr. Nilles is a Movement Disorders Fellow at the University of Calgary, Department of Clinical Neurosciences, Psychiatry, Pediatrics, and Community Health Sciences. She came to the University of Calgary from the Sorbonne University in Paris, France, where she did her residency in neurology and completed a Master of Science in Integrative Biology and Physiology, specializing in neurosciences. I hand it to you, Dr. Nilles. Thank you.
Thank you, Julian, for this introduction, and good morning, everyone. Thank you for joining me today. Let me just share my screen. I think now you can see it full screen. Please tell me if it's not the case. So today I'm going to present to you, as Julian said, the familial risk of Tourette syndrome and OCD for about 50 minutes, and then we will have a time for questions and comments. So the objective of this webinar first is to compare the heritability of Tourette syndrome and OCD. Then I would like to review with you the different techniques and studies used to assess this genetic risk. So I'm not going to go into too many details, but I think it's interesting and important to have certain notions. And then we will discuss how to communicate this risk to patients and what the clinical implications are. So, as you may probably know, parents frequently ask if Tourette syndrome and OCD are genetic diseases. And they also wonder why do their children have several mental health disorders. And these are both very relevant questions because both Tourette syndrome and OCD are frequently associated along with other disorders that I represented here under the neurodevelopmental umbrella. So there are OCD, Tourette and tic disorders, ADHD, behavioral disorders, mood and anxiety disorders, substance abuse, and sleep disturbances. So if all these disorders can be frequently associated in one patient, they are also found in relatives. And so when we ask patients if their relatives have the same disorder as they do, 47% of patients with Tourette syndrome say that there is a tic family history. Interestingly, tics are also often undiagnosed in the family. And for OCD, the prevalence in first-degree relatives has been found to be between 10 to 12%. So genetic studies have investigated the mode of transmission of both disorders in recent decades, and that is what we are going to see together. So first, some definitions. Tourette syndrome, as you know, is the association of at least one vocal tic and at least two motor tics for more than 1 year with an onset before 18 years old. Tourette syndrome is a very common disorder. It is found in 1% in school-age boys and 0.77 in adult children. Tourette syndrome has a clear male predominance as the sex ratio is 4 males to 1 female, and the peak for the beginning of tics is 6 years old on average. Obsessive compulsive disorder is defined by the DSM-5 criteria by the presence of obsessions, compulsions, or both. And these symptoms have to be distressful, right, to meet the criteria of OCD. So they have to be time-consuming or cause clinical distress or have an impact on the functioning. They have to be a disorder on themselves, meaning they must not be secondary to another disorder or a drug. So OCD is also a very common disorder with a lifetime prevalence between 1 to 3%. And the sex ratio is not equal, but interestingly, it varies over time with mostly males when OCD has an early onset, and then a female predominance in adolescence and adulthood. There are two peaks for the symptoms of OCD, one during adolescence between 13 and 18 years, and then during early adulthood. So why are we presenting OCD and Tourette syndrome during the same webinar? Well, they share a lot in terms of symptoms and also epidemiology. First, just simply, they both consist in repetitive patterns of behavior, so tics and compulsions. And in both disorders, there is specific subtype for the other one, meaning in tic disorder, there is a specific tic called tic-related compulsive behavior, which is a subtype of tic looking like compulsion. So for example, it could be a touching tip, a tapping tic, or an evening up phenomenon. So typically, the children will say, I have a tic where I touch my right thigh, and then I have to touch the other thigh. So this is a tic with a compulsion in it, I would say. And also in OCD, there is a subtype of OCD called tic-related OCD or Tourettic OCD, where the compulsions look like tics. In Tourette patients, OCD is present in 50%, and subclinical OCD, meaning obsessive compulsive symptoms, but which do not meet criteria for OCD, are found in 66%, which is a lot. So it makes sense to analyze both disorders together, right? Also, females with Tourette syndrome are more likely to have comorbid OCD, so maybe a sex dimorphism in this. And both disorders are associated, as I mentioned before, with other conditions like anxiety, major depressive disorder, and ADHD or autism spectrum disorder. So my first part is going to focus on the heritability of Tourette syndrome and OCD. And so the first type of studies which search for to specify the genetic risk in Tourette syndrome are family and twin studies. So even quite early, right, in 1985, there were studies on twins. So they looked at the concordance between twins, concordance being defined by the rate at which a pair of individuals share a diagnosis. So they looked at monozygotic twins and dizygotic twins. So monozygotic twins are twins who share 100% of their genome. They come from the same egg. And dizygotic twins have shared 50% of their genome, like regular siblings. And so if Tourette syndrome is a heritable disorder, they would expect that the concordance rate in monozygotic twins is higher than the concordance rate in dizygotic twins. And so this is what they found in this first study on 43 twin pairs. For Tourette syndrome, the monozygotic concordance was 53% higher than the dizygotic concordance of 8% only. In any tic disorder, not just Tourette syndrome, also the monozygotic concordance was at least twice higher than the dizygotic concordance. Then in other large family studies, they looked at the prevalence, the recurrence rate, right, of Tourette syndrome in relatives. They found that in females, 5% of the relatives had Tourette's and more than 10% of males. Another study found that first-degree relatives had a 10 to a 100-fold increase in risk for having Tourette's. Two more studies, more recent and on quite large samples. So this one on 2,600 monozygotic pairs and 3,700 dizygotic pairs found the same result as before, that the monozygotic concordance was at least higher than the dizygotic concordance in twins. And so all these studies could allow calculating the heritability. So what is heritability? I found this definition easy to understand. It is the estimate of how well differences in people's genes account for differences in their traits. So this study in 2015, I'm going to talk a lot about this registry because this is a very large sample in the Swedish National Patient Register, which allowed to know quite a lot about genetics in neuropsychiatric disorders specifically. And so on 4,000 patients with Tourette, they found that the heritability of tic disorders was 77%, meaning 77% of the clinical variance was attributed to genetic factors. So the remainder being most likely due to environmental factors that we will talk about at the end. And so what about OCD? So family studies also showed a recurrence risk among first-degree relatives for lifetime OCD higher than the prevalence of overall population. So all these studies estimated a quite wide range from 6 to 55%. And interestingly, the risk was also higher in relative for just obsessive compulsive symptoms and behavior. In twin studies, the results are quite similar than for Tourette, since the OCD concordance rate in twins was higher in monozygotic twins, 52%, than in dizygotic twins, 21%. The same pattern was observed for obsessive compulsive symptoms, with concordance rates twice higher in monozygotic twins than in dizygotic twins. This nice review of Majani et al. In 2021 kind of summarized all the studies which allowed to calculate the heritability of OCD and obsessive compulsive symptoms. So here you have all the studies which have indeed different methodology and assessment methods, but this is obviously still useful. And you have here the circle one is the obsessive compulsive symptoms, and the square ones are the OCD estimates. And so even if the results are quite variable, we could say, it seems like the heritability of OCD could be estimated around here. And this study on the Swedish adult twins I was telling you about earlier found that a reliable estimate would be 42 to 65% for OCD. We saw together that Tourette and OCD were both heritable disorders, and that notably the familial aggregation of Tourette syndrome of 77% was substantially higher than the familial aggregation of OCD of 42 to 65%. Now I'm going to review with you how genes might contribute to Tourette syndrome and OCD, with a little bit of history of the genetic studies. So at first, let's say in 1990, the hypothesis was that Tourette and OCD were autosomal dominant diseases, meaning they would be due to one mutation in one gene in one chromosome. So the first studies which looked for that are the linkage studies. These studies looked for a correlation between a trait, so a disorder, and a chromosome region in families. So they had several candidates looking, but no gene or causal mutation of major effect has been discovered. This locus has been suggested to be responsible for syndrome on chromosome 15 in 9 relatives with Tourette syndrome, but it was not reproducible in recent studies and additional Tourette cases. So the hypothesis following these kind of studies where maybe some families carry a heavy load of common risk variation, or maybe somewhere non-coding risk variant have high penetrance that we will review later. And so the second type of studies which looked for to prove that Tourette and OCD were indeed autosomal dominant diseases were candidate gene association studies. So these studies assess allele frequencies within populations. So the first study was looking in families, and this one is looking for these frequencies in overall populations. And so they look for what we call single nucleotide polymorphism, SNP. So this is simply you know how DNA is composed of different nucleotides. And so this is just one nucleotide replaced by another. We call this SNP. And so in Tourette syndrome, they had some candidate genes to look for based on the physiopathology of Tourette and the response to treatment, right? So for example, they knew at that time that Tourette patients could respond to antipsychotics, which blocks the dopaminergic transmission. So they looked for maybe mutations in dopamine receptor and transporter genes. So based on this kind of reasoning, they looked for SNP in neuroadrenergic transcript, genes affecting neurotransmission, and genes involved in neurodevelopmental, neuroendocrine, and metabolic functions. Unfortunately, no significant reproducible findings have been found for Tourette. And so what about OCD? So same for Tourette, we had a lot of candidate gene for this to look for, based again on physiopathology. And OCD was associated with polymorphisms in several genes, serotonin genes, dopamine genes, glutamate genes, and genes involved in immune and wild matter pathways. Unfortunately, the results were quite mixed, and these findings were not often replicated, which is necessary to call one variant a genetic risk for the disease, right? So this may be due to small sample sizes or just to methodological difficulties to obtain a statistical significance. Then they started to think, okay, maybe Tourette and OCD are not autosomal dominant diseases. Maybe they are polygenic diseases, meaning several genes are responsible for the disease. And so one study looking to prove this hypothesis was the copy number variant studies, CNV. So what are CNV? CNV are structural variation. So for example, duplication, deletion, insertion that affect large segments of DNA. So for example, here you have a chromosome, and in different individuals, some segments of this chromosome can be deleted or duplicated. And this region can encompass genes and lead to dosage imbalances. And it is a rare phenomenon, but it is something to look for, especially in neuropsychiatric disorders. So for Tourette's, several studies found a significant excess of rare CNVs, so potentially pathogenic. And interestingly, these CNV were previously implicated in other disorders, schizophrenia, autism, and ADHD, which kind of makes sense since we saw that clinically there was this overlap. So the fact that they found an overlap in genetics makes sense. In OCD, it was a bit different. The overall rate of copy number variance burden did not differ between OCD patients and controls. They did find a potential higher frequency of rare CNV affecting brain genes, which were previously linked to other disorders, Tourette syndrome, neurodevelopmental disorders, but it was not significant. It was just a tendency. So, yeah. Another study which helped a lot the field was the GWAS studies that you may have heard of, the Genome-Wide association study. These kind of study look for a correlation between genetic variants, so common SNP with a frequency usually higher than 5%, with a trait or disorder in different individuals. So this is case control studies or family-based. They look for a frequency of one allele in cases and in controls. And then they compare the frequency between the two using an array. And they look, they analyze if there is a statistical difference. And so GWAS studies are a powerful tool to detect genetic risk factor at the human genome-wide level. So in Tourette's, the first GWAS study of common genetic variants did not find any significant loci, but probably it was due to insufficient sample size, because for this study, you need very large samples, and it was the first one. But even the largest and most recent GWAS study did find one significant locus in this gene, but it failed to replicate in another cohort. Interestingly, quite a recent study by Domenech et al. Found that polygenic risk scores could not only predict the Tourette syndrome affected status, but also the tic severity, suggesting that a high burden of genetic risk factor could lead to a higher severity of tics in patients, which is interesting. In OCD, 6 GWAS studies and 1 meta-analysis were done. Until 2021, no genome-wide significant loci was identified. And in 2021, one locus in the gene encoding PTPRD protein was associated with obsessive compulsive traits. So what is this gene? So this is a gene which is involved in several cellular processes, but also in the, which is involved in the glutamate neurotransmission. And this is interesting because OCD is thought to be related to abnormalities in the glutamate neurotransmission. So this is a lead. Then we have the WES studies, the Whole-Exome Sequencing studies, using the next generation sequencing that you may have heard of. And these sequencing approaches sequence all coding regions of the entire human region. So you may know this, but in the genome, there are the vast majority of the genome are non-coding, and the minority or coding region, meaning that the genes will lead to the production of proteins. And so this is only these kind of regions that the WES studies are looking for. So they try to identify where variants, insertions and deletions, and single nucleotide variants. This is a fast and cost-effective method. And within psychiatric genetics, it has been used to detect where coding variants that are near absent from the general population. So in Tourette syndrome, several WES studies have been performed since 2011. And de novo damaging variants, so potentially pathogenic, were found in 400 genes. And they're thought to be contributing to the genetic risk in 12% of cases. We also identify likely risk gene for Tourette syndrome, but none of the results have been confirmed in larger samples or supported by functional studies. So they are not yet classified as Tourette syndrome risk gene, but they do support the hypothesis that Tourette syndrome is due to polygenetic factors. In OCD, this is a nice study which looked for de novo damaging variants between OCD patients in red and controls in blue. So this is all the different kind of de novo variants that that they looked for, and you can see that OCD patients mostly have more mutation rates in these variants than controls in every category. And so these variants in 335 genes are thought to contribute to the risk of OCD in 22% of cases. And again, there was a significant overlap between these genes and the genes previously reported in Tourette syndrome and autism. The last study, which helped us to understand the genetic risk of these disorders, is the whole genome sequencing, WGS. So as I said before, the WES study, they just looked for coding region of the genome, but the WGS completes the characterization of common and rare variation, all variants detectable in array and WES data, and where non-coding SNV and insertions and deletions. They also look for copy number variants that are too small to be reliably detected in array data. But these studies are quite difficult because so you have a lot of data and it is quite costful. So we saw together that Tourette syndrome and OCD were complex, highly heritable, and polygenic disorders. So why can't we find a responsible gene? So several reasons to that. First, both disorders have a phenotypic complexity. There is a clinical and genetic heterogeneity of Tourette and OCD. And the studies so far had limited sample size compared to other psychiatric disorders like schizophrenia. Also, the samples are mostly European, which could be a bias. The previous genetic technologies were limited by statistical defaults. And also, these are costful methods specifically for the WGS. So I would like just to go a little bit further into the genetic risk in comorbid Tourette and OCD, since we kept saying that there was an overlap. So about this co-occurrence of tics and OCD, family studies have been done. And so Tourette syndrome and OCD have been found to have a significant genetic correlation of 92%. Also, they did a twin study in twins with tics and OCD. And they found, again here, that the monozygotic concordance rate was twice higher than the dizygotic concordance rate in this Swedish cohort. So another nice study about the genetic correlations across psychiatric phenotypes. Very large GWAS meta-analysis which actually did not just look at the psychiatric, but also neurological disorders in general. And so you can see that OCD was correlated with Tourette syndrome. It was significant. OCD was also related to other disorders like depression and anorexia nervosa. So we may think maybe these two disorders have the same underlying genetic factors, or other hypothesis, OCD may be an alternative phenotypic expression of Tourette syndrome susceptibility genes, or a more severe expression having a greater genetic loading. So these are hypotheses for now. Further studies I told you a little bit of copy number variance. They looked for abnormalities of this in Tourette and OCD. There was nothing significant, but a tendency to threefold increase burden of large deletions in patients with both disorders. And 50% of these deletions were found in a locus already identified to have a role in developmental delay and autism spectrum disorder. I would just like to say a word about this subtype of OCD called tic-related OCD or Tourettic OCD, which has specifics, first clinically, since these disorders has an earlier age of onset. Males are predominantly affected compared to the other types of OCD where females are mainly affected. And there is a higher rate of symmetry and sexual and aggressive obsessions. And they wondered if this subtype of OCD had a higher heritability. And they proved it in this study, looking for the risk of OCD in relatives of individuals with tic-related OCD, which was very much higher than the risk of OCD in relatives of other OCD patients, risk of 10 versus 4. Last study, which I think it's nice to see. Again, they looked for genetic correlations between Tourette and other psychiatric disorders from a GWAS summary statistics. So here you have the genetic correlation with Tourette syndrome and here different psychiatric disorder. And you can see that this is quite a large cohort. So for example, Tourette syndrome was correlated to ADHD with genetic correlation of 0.21. And with OCD, the genetic correlation was the highest with a 0.42 genetic correlation. Just to say, even if OCD and anorexia were related before, the genetic correlation between Tourette syndrome and anorexia nervosa was very low. So I mentioned some unequal sex ratios in both disorders. So I just wanted to discuss a little bit about maybe a sexual dimorphism in genetic risk. So for example, in Tourette syndrome, so there is a male predominance, and so they looked for differential heritability by sex. And in this twin study from Sweden, they found that the heritability in boys was 39% versus 26% in girls. But later reports in this nice review showed that actually it did not differ between males and females. In OCD, we saw that there were sex differences in age of onset, but also in clinical presentation with the Tourettic OCD subtype. And again, discordant results from twin studies. This twin study showed a higher correlation in males only, in monozygotic twins compared to dizygotic twins. Whereas the female correlation between monozygotic and dizygotic twins was similar. But there was no genetic sex differences in another twin study in the Netherlands. So yeah. This nice study also analyzed the sex-specific genetic architecture of OCD. They did not find sex-specific single nucleotide polymorphism, but they did talk about 2 genes associated with OCD only in females. And in these 2 genes, again, we find 1 gene which is involved in the glutamate neurotransmission. So now I'm going to talk about the clinical and therapeutic implications of knowing about this genetic risk. So first, it is helpful to inform the patients and the families, right? It is always helpful to give reliable information and to fight against certain preconceived ideas that they may have. So it's important to say that OCD and Tourette are complex disorders with polygenic risk. And so this is not, as sometimes parents ask us, this is not the fault of one parent or the fault of anyone. Also, OCD is not secondary to trauma, but is influenced by life experiences, like many other disorders. And just remind that Tourette and OCD are very common disorders. As we said, 1% in school-age boys for Tourette and 1 to 3% for OCD. And just remind that the understanding of the genetics of Tourette and OCD remains incomplete, and there is a lot to do and to discover. So advantages of knowing about the familial risk. So first, it is helpful to provide a mechanism for the comorbidities. So we suggested that, the researchers suggested that there was a common genetic risk between both disorders, and this is very helpful to go further into research. Also, I think it's also useful to know about this genetic risk to diagnose early OCD, Tourette, and other comorbidities in the patients, but also in siblings. So knowing this risk is knowing what to look for in the patients. For example, in clinics, when we have a Tourette patient, we also ask for all the other neurodevelopmental disorders. And I guess it is the same for OCD. Also, it is not uncommon that when we diagnose tics or OCD in a patient, that the parents ask themselves, oh, maybe I have the same symptoms, right? And it is important, this early diagnosis, to improve the quality of life. Tourette syndrome, in studies in Tourette syndrome, actually, the quality of life was more impaired by the comorbidities like OCD than by the tics themselves. So it is crucial to make this diagnosis. Another example, more focused on OCD, this early treatment is important for a positive outcome, as OCD has unfortunately a long duration of untreated illness, about 7 years on average, and late interventions are related to poor therapy response, which is a shame. An example of novel therapeutic targets thanks to genetics, even if it's really at the beginning, I would say. So as I mentioned, glutamate is involved in OCD, which has been associated to a hyperactivity of the corticostriatal-thalamocortical circuitry. And so they looked for glutamate modulating drugs as potential treatment options for OCD. So, for example, memantine has been shown to have positive effects in OCD. Other drugs like anticonvulsivants, lamotrigine and topiramate, but also riluzole and ketamine, which has a rapid effect of action, are thought to be maybe also effective. But obviously, this is really the beginning of this kind of research, and further trials with randomization and control by placebo in larger study populations are necessary. Also, this is this is a field where there is also a lot to do, and there is a lot of hope. Genomics and therapeutic in OCD, for example. So this field, the pharmacogenetics, I put the whole definition here because I think it's helpful. It defines the genetic variants that influence drug metabolism, delivery, affinity to receptor or transporters, which may contribute to the prediction of drug efficacy or toxicity. And so this is very helpful, for example, for OCD, because 1/4 of patients do not respond to treatment with either SSRIs or cognitive behavioral therapy. So I think this is good that we look for biomarkers to help us identify these patients. So one study, early study, found that one locus in one gene was associated with treatment response to SSRIs, and it could be helpful in the future in real life. So I would say that having a greater knowledge about these specific genetic risks for both disorders not only will help us understand the underlying biological mechanism, but also provide opportunities to develop new animal or cellular models for research, but also identify new biomarkers for diagnosis, looking for individuals with a high risk factor of the disorder, and for prognosis, looking for individuals who will respond or not to treatment, and so allowing testing new therapeutic avenues. So maybe you are wondering, are there non-genetic risk factors? And so there are. First, in the environment, pre and perinatal birth risk factors are both related to Tourette and OCD. So, for example, an impaired fetal growth was associated with Tourette syndrome. You have here the hazard ratio. So you can see that it is elevated for Tourette and OCD according to the birth weight. Also, being premature, it's associated to Tourette and OCD. The breech presentation, being born through a C-section. Maternal smoking, even if there is variable results, but it might be associated with a dose response manner. And the ABGA score, which is a score for distress at birth, was associated in OCD only. Just a word about stressful life events in OCD. Stressful life events meaning childhood trauma or amount of life events only. Simple studies showed that 25 to 64% of patients reported stressful life events before the onset of OCD. Other studies found that these stressful life events in the past 12 months were associated with a 21% increased likelihood of OCD. And this specific risk factor could be could depend on sex, since women typically reported more stressful life events before the OCD than males. Autoimmunity is a trendy topic, and it has been also associated with Tourette and OCD. This is, I think, nice graphs which represented the risk of autoimmune disorder, so all autoimmune disorder confounded, in the patient with OCD or Tourette. In red, you have the first degree relatives and their risk to have autoimmune disease as well, and then second degree relative and third degree relative. And so first, you can see that there is an increased risk of autoimmune disorder for patients with OCD for 43% and with Tourette 36%. And also interestingly, you can see that the risk of autoimmune disease is also present for relatives and seems to decrease over the degrees of relatives. So we discussed the genetic risk factor, the environmental risk factor, and finally there are epigenetic risk factors. In a word, epigenetic is the ongoing regulation of gene expression. So it allows the reprogramming of the genome in response to environmental inputs. So just to cite only two. It can be, so you have the chromosome, and then you have all the epigenetics represented, not all, but some of the epigenetic processes that can happen. So structural variation of the chromatin or chemical modification like DNA methylation. And this has an impact on how genes will be translated. And modification in all these processes have been found to be a valuable hypothesis for several neurodevelopmental behavioral problems. So in Tourette syndrome, the first epigenome-wide association study, GWAS, of tic disorders, was interested in the DNA methylation. No methylation sites reached genome-wide significance, but several of the 10 top-ranking probes were in or nearby genes previously associated with neurological disorders. In OCD, several studies have been done with variable results. No significant differential methylation, but there was an association between DNA methylation in two regions and baseline OCD severity, treatment effect, and responder status, which is very interesting for the future. So there is definitely a need for further investigation because all of these studies are very recent and are at the beginning. So I would say that the phenotype OCD and Tourette syndrome are due to genetic risk factor, polygenic risk factor, with most likely contributors from common and rare variants that we have to identify more precisely with larger sample sizes and a more robust statistical methodology. And also non-genetic risk factor, including the environment and the epigenetic. So for the future, I would say that what's ahead of us is studies with larger sample sizes, as I said several times. Currently, 4 large collaborative groups are joining efforts and resources for Tourette syndrome genetic research, looking for more variants. Also, epigenetic research is a big part of the future. It will look for link between genomic variations, environmental exposures, and diseases outcomes. And finally, it would be interesting to understand a bit better the mechanism behind some specific clinical features, and I'm thinking of sex differences. So I would say that the takeaways of this talk are that Tourette syndrome and OCD are both complex, heritable, heterogeneous disorders with a higher heritability of Tourette syndrome, 0.77 than OCD, 0.42 to 0.65, and that most likely there is a genetic correlation between Tourette and OCD, suggested that they are due to the same underlying genetic factors that remain to be precisely identified. It is useful to communicate this genetic risk to the families as clinicians and to understand it better as researchers. Other risk factors will include the environmental risk factors with pre and perinatal risk factors, as I mentioned, and the epigenetic, which is an open door for research.